Hey, physiology students. I don't know why the picture isn't so clear. It's a little blurry today. But anyway, this is going to be Friday's lecture, which is the end of chapter 2, uh, part 1, cell metabolism. Okay, so I'm going to go ahead and share the screen and get to the PowerPoint. Okay. Okay, so this is what we've got left. So this would be a short lecture. Yay! So we had talked last time on Wednesday about lipid metabolism. So if I go back one slide, let's make this bigger here. There we go. So if I go back, we had talked about lipid metabolism. We got into ketosis, ketogenesis, all of that, okay? So what we've got left, the last thing we have to talk about is amino acid metabolism. So let's recap real quick here. We started out at the beginning of this chapter talking about glycolysis, which is used to make ATP. And then we talked about glycogen metabolism, and both of those are carbohydrate metabolism pathways. Then we talked about lactic acid metabolism. Then we talked about lipid metabolism, and now we're up to amino acid metabolism. Okay, so you metabolize amino acids anytime that you consume protein. So you absorb amino acids across the lining of your small intestine, and then these amino acids are floating free in your bloodstream, and your cells take it up and then process it to make it into your own cellular protein. Okay, just a little bit of vocab here. You probably had this information in intro biology, but there are essential amino acids. Those are essential because you have to consume them in your diet. Your body can't make them. And those are relatively few amino acids that our bodies can't make. Non-essential amino acids are the ones our body can make, and our body can make most of them. Okay, so let's say you consume a lot of protein, okay, or you just happen to have excess amino acids in your bloodstream from consuming a lot of protein. That's one way you can have those in your bloodstream. Another way you could have those in your bloodstream is if someone is going through the phases of starvation, where they've used up all their carbohydrates, they've used up all their lipid stores, and then the body will turn to the protein stores, and your body will break down protein to fuel its energetic demands. So excess amino acids, I'm symbolizing just AA, get converted by the liver into pyruvate or other acids, okay? So the liver, again, is saving you from metabolic acidosis, just like it did when there were lactic acid in the bloodstream from muscle activity. The liver cleans that acid from your blood and turns it into something useful. Remember, with the Cori cycle, lactic acid in the blood is taken up by the liver, and the liver converts it into pyruvate. And from pyruvate, if you've been practicing your glycolysis pathway, pyruvate goes through conversion, becomes acetyl-CoA. Acetyl-CoA goes through the Krebs cycle, produces lots and lots of hydrogen atoms. Those get shuttled along the electron transport chain, and bingo, bango, you've got a lot of ATP. But pyruvate could be turned into all kinds of other things as well. If you remember, in the Cori cycle, liver took lactic acid, turned it into pyruvate, and then from pyruvate, it went backwards through glycolysis to make glucose. So the liver can do this as well, all right, starting with amino acids. So as long as you've memorized that glycolysis pathway, there's a lot of repeat. Lactic acid can be converted to pyruvate. Pyruvate goes backwards through the glycolysis pathway. If you have to break down lipids, you go from triglycerides to fatty acids, and then fatty acids can be converted into acetyl-CoA, and then acetyl-CoA goes backwards through conversion to produce pyruvate, and then pyruvate goes backwards through glycolysis to make glucose. So as long as you've got pyruvate, you can go one way in the pathway, or you can go another way in the pathway. So let's see what the liver does with this pyruvate. Okay, so that pyruvate, if there's oxygen present, will go through conversion to make acetyl-CoA. And the acetyl-CoA jumps into the Krebs cycle to start ATP production. So Krebs cycle produces hydrogen ions, and then they go through the electron transport chain. So I'm not going to repeat all of that here, but that's what could happen. Here's the other thing that could happen. You can convert the pyruvate into fat, okay? Remember making fat. In fact, I'm going to go backwards in the PowerPoint real quick here. Oh, lipogenesis. Okay, here it is. Once you have pyruvate, okay, it can go down one pathway or backwards, okay? So that's what the liver can do. If you've memorized this pathway, once you've got pyruvate, you can make fat from it, okay, by following this slide. So let's go back to here. So that pyruvate can be converted into acetyl-CoA. Acetyl-CoA can be used to make fatty acids that can convert it into storage fat known as triglycerides. That's lipogenesis. The liver is not going to waste these amino acids. It's going to convert it into something the body can use, and in the case of fat, store it in case the body needs it. Okay, if the liver has converted amino acids into pyruvate, a quick step would be go backwards through glycolysis and turn pyruvate back into glucose. That is known as gluconeogenesis. All right? Okay, let's see what else happens. Now, what the liver cannot convert into useful things like fats or glucose or making glucose into glycogen, what the liver cannot possibly process, it's going to process to get rid of from the body. So what it will do, it will convert those excess amino acids, let me go back, to urea. And urea goes in the bloodstream. And then the urea is excreted by the kidneys as ammonia, and that's what comes out in your urine. Okay, so urea is a nitrogen-based or protein-based byproduct, as is ammonia. Okay, now the urea floats around in the bloodstream. In order to get that urea excreted by the bodies, you have to have functional kidneys in order to excrete that urea as ammonia. The kidneys are supposed to excrete urea, blood urea nitrogen, at a constant rate. And if your kidneys aren't getting rid of this urea, the blood urea nitrogen, it's probably an indicator that the kidneys aren't functioning correctly. So here we have blood panels. Most typical blood panels look for cholesterol, look for fatty acids, triglycerides, glucose. They also look for blood urea nitrogen. That is a byproduct of amino acid metabolism by the liver. And so that urea is in the bloodstream. Now, the normal BUN is supposed to be 10 to 20 micrograms per deciliter. If you have high blood urea nitrogen, that is usually indicative of kidney failure. That's called azotemia. That means your kidneys aren't clearing the urea from your blood. It indicates a problem. So as nurses, you'll learn so many normal values, okay, especially for things that you'd look for in a common blood panel. I guarantee you, if you've ever had your blood chemistries done, you've had a BUN run on your blood. Okay, so let's move on to our blank flow diagram, the amino acid metabolism pathway. What I've got is going down here, back up, going down here, what the liver does right here converts the extra amino acids that it can turn into useful things. First, it starts by converting into pyruvate, and then from pyruvate, you can go anywhere in the cycle, even go backwards. So if you want to make ATP, pyruvate goes through conversion to make acetyl-CoA. Acetyl-CoA enters the Krebs cycle, pumps out a bunch of hydrogen ions. Those hydrogen ions enter the electron transport chain, and you produce 30 to 32 ATP. Okay, great, that's that process. What else can pyruvate be changed into? Now, the previous slide I told you, for one thing, you can turn pyruvate into fat, and that is lipogenesis. Instead of starting with glucose, you started with amino acids and then to pyruvate. So pyruvate can be changed into acetyl-CoA. Acetyl-CoA can be converted into fatty acids, and fatty acids get converted into white fat known as triglycerides. If you go backwards through glycolysis, if pyruvate goes backwards through glycolysis, what do you make? Glucose, gluconeogenesis. All right. So I don't have it filled in there, but let's go ahead and fill it in for you. Okay. Oh, that's the blank flow diagram, I forgot. But when you're looking at the PDF of this PowerPoint, you'll be able to click here for the blank flow diagram. It looks just like this, and here for the key. It's also posted on the online syllabus. Okay, so that's amino acid metabolism. Let's talk about a problem with, in another slide, we'll talk about problems with amino acid metabolism. But first, I want you to think. We've talked about lots of things that when your body metabolizes them and when they're floating around in your bloodstream, they will drop your blood pH, and that's called metabolic acidosis. So, hint, hint, if it ends in the word acid, it's going to make your blood pH drop. Okay, so think of some molecules like what we just talked about, amino acids. Okay, so I can back out of this and answer this one. Okay, so amino acids, it's got the word acid in it. All right, so you know it can drop the blood pH. What's something else? How about in a lipid pathway? Fatty acids. Okay, got the word acid in it. What is a type of fat that I mentioned that some people, when you're metabolizing and using these types of fats, they can drop your blood pH, and some people do this on purpose because they're trying to cut body fat by avoiding carbohydrates entirely. If you said ketones, you're right. Ketones are acidic and they will drop your blood pH. What is the name for the drop in blood pH? That is metabolic acidosis, but it's got its own name based on ketones. What is that word for the drop in blood pH by using ketones for energy? So, ketoacidosis is what that's called. Okay, and then there's one more thing. From normal skeletal muscle activity, it will produce this byproduct that causes the burn and the muscle burn. Lactic acid. Okay, so and there's more things your body can metabolize because there's a lot more acids in these pathways I've talked about, but those are the ones I want you to focus on. That's what I've covered in this lecture. Okay, problem processing amino acids. It's very common. It's called phenylketonuria. All right, PKU. It's a genetic condition, and we'll look at the online app in a second. But what happens is people have a mutation in a gene for the enzyme that you're supposed to have called phenylalanine hydrolase. So it ends in ASE, it's an enzyme. It hydrolyzes or breaks apart the amino acid phenylalanine. If you have the mutation in the gene, you don't have this enzyme, you can't break down phenylalanine. Well, how common is PKU? So common that if you ever look at the ingredients and facts, sugars, carbohydrates, salt, all that you see on the information part of any product, if it contains phenylalanine, there will be a warning. So it says ingredients: dextrose with maltodextrin, aspartame. Oh, aspartame has phenylalanine in it. Phenylketonurics, people who have PKU, this contains phenylalanine. That means warning, warning, don't eat this product, because what will happen is this. You need the enzyme phenylalanine hydrolase to metabolize phenylalanine. Without the enzyme, the body converts those phenylalanine molecules into phenylketones, which are highly toxic. All right, how toxic? It can cause seizures and possibly death. So the treatment is to avoid phenylalanine in the diet. So nutrition labels, if the product contains phenylalanine, they'll have PKU. Warning, warning, this has phenylalanine. Okay, all right. So review, all right, from Wednesday to today. On Wednesday, we talked about lactic acid metabolism, lipid metabolism, and today, Friday, we talked about protein metabolism. Okay, so this ends the metabolism chapter, chapter 2, part 1. So on Monday, we'll start chapter 2, part 2, which is cell transport, things like passive and active transport. All right, so have a good weekend, and I'll talk to you on Monday. Okay, and we will end.